Hello, secondary 2 students. Today we will talk about cellular respiration. But first we have to know what are the difference between the gas exchange and the cellular respiration. Gas exchange meaning exchange between the oxygen and carbon dioxide without being oxidized or without using the oxygen to make anything inside the cell, just exchange of the gases. But the cellular respiration means extracting the energy from the bones of the glucose molecule or the food molecules and use it to form energy currency of the cell or something called ATP molecule. So this lesson is talking about how to extract this energy from the glucose molecule or how to form the energy by the respiration. It occurs by 3 steps. One of them is called, or the first one is called the glycolysis, followed by Krebs cycle and electron transported chain process. Glycolysis occurs inside the cytosol or the cytoplasm of the cell, and the second 2 steps will be formed inside the mitochondria. So let's start with the structure with ATP molecule. It's like the structure of nucleotide. It's formed of ribose sugar, nitrogenous base called adenine, and the only difference between it and the nucleotide is that it's contain 3 phosphate groups, so it's called ATP. If we break down this bond between these 2 phosphate groups, it will form ADP or adenosine diphosphate. But if the energy consumed to form this bond, another phosphate group will be combined with ADP, forming ATP molecule. Each ATP molecule, after breaking down this bond, will release from 7 to 12 kilocalorie per mole. So the energy produced from glucose molecule oxidation will be 38 ATP, and each ATP molecule will form these numbers of calories from 7 to 12 kilocalorie. So we can calculate if you burn or if you oxidize one glucose molecule, it will release from 7 to 12 kilocalorie per mole, multiply 38 molecules of ATP. Let's start with the glucose oxidation by the first step, which is called glycolysis. But before this, here we have the equation for the glucose oxidation. Start with the glucose and oxygen. Here is the arrow, miss the oxygen gas. Oxygen will burn the carbohydrates or the glucose to form 6 atoms or 6 molecules of carbon dioxide, 6 molecules of water, and 38 ATP. We can follow the steps to get this result: carbon dioxide, water vapor, and ATP. The first step, which is glycolysis. Glycolysis starts with the glucose. In turn, glucose will change into glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-biphosphate, ends with the pyruvic acid, phosphoglyceraldehyde or BGAL, then pyruvic acid. The net of this or these changes will lead to formation of 4 ATP molecules and 2 NADH. What is NADH? NADH is NAD, which is coenzyme inside the mitochondria or the carrier of the hydrogen or receptor for the hydrogen that will be reduced to form NADH to carry this H to the last step, which is electron transported chain. So if we calculate the amount of ATP produced, here ATP changed into ADP, so we can break ATP to form ADP and release 1 phosphate group, which is bind to this glucose, forming glucose 6-phosphate. Also here we can change ADP into ATP and also using phosphate. So the net of the glycolysis is formation of 2 ATP molecules and 2 NADH. The second step is the Krebs cycle, and the Krebs cycle starts with the intermediate step, which in turn produces carbon dioxide and also will produces 2 NADH. In this step, pyruvic acid will combine with something helper or coenzyme called coenzyme A. After that, the pyruvic acid, by the help of coenzyme A, will enter the Krebs cycle, forming acetyl-CoA, and the CoA enzyme will be separated again to searching for another pyruvic acid to combine with. Acetyl-CoA is 2 carbon atoms compound, which in this stage also amino acids and fatty acids in food will join the Krebs cycle in the form of acetyl-CoA, and 2 molecules of carbon dioxide will be produced. Let's start the Krebs cycle. Here we start with 2 acetyl-CoA. The first compound produced inside the Krebs cycle is the citric acid, so we can call the Krebs cycle a citric acid cycle also. And we have 4 intermediate compounds after the citric acid. We have oxalic acid, acetoacetic acid, ketoglutaric acid, succinic acid, and malic acid. All of this intermediate compound, to change from one to another, will produce NADH and will produce FADH and will produce ATP. Here in this step, or this cycle of citric acid cycle, will be repeated 2 times. One for the acetyl-CoA and the other for the second acetyl-CoA. So if we calculate the amount of NADH produced in this cycle, we can multiply it by 2, one for each acetyl-CoA. So the net or the result of the Krebs cycle that we have 3 NADH, we have and multiply it by 2 because this cycle repeated 2 times. We have 1 direct ATP molecule that we will multiply it also by 2 because this cycle repeated 2 times. Also we have 1 molecule of FAD multiply 2, so we have 2 FAD molecules. So this Krebs cycle ends in formation of 6 NADH, 2 FADH2 and 2 ATP molecules. We can call ATP molecules as a direct energy resource, but NADH and FADH are called indirect source of ATP. The third step of glucose oxidation is the electron transported chain. What will happen here in the electron transported chain? As you know that the inner membrane of the mitochondria contains molecules called cytochrome. This cytochrome will split the hydrogen which are carried in the NAD and FAD into hydrogen proton and electrons. And these electrons will pass from higher energy level inside the cytochrome to the lower energy level, releasing energy. This energy will be used by ADP and phosphate to form ATP molecule. So if we take the hydrogen of the NAD, it will release energy enough to form 3 ATP. So we can consider that each NADH will be produce 3 ATP molecules, and each FAD molecule has H that will split into electrons and the protons, and this electron will produce enough energy to form 2 ATPs. So we can also consider that each FAD H2 will produce 2 ATP molecules. If you can calculate the 38 ATPs that are produced from all these steps, in the first we have 2 ATP molecules direct from the glycolysis, and in the Krebs cycle we have also 2 ATP molecules direct. And each NAD and FAD produced during the 3 steps will be transferred into ATP by equalizing the NADH into 3 ATP and FADH into 2 ATP. So we calculated here the ATP molecules that are produced, the carbon dioxide molecules that are produced in the step between the glycolysis and Krebs cycle, and the final stage is formation of water vapor. The protons that are released from splitting of hydrogen atom and the electrons produced will join half oxygen molecule, which is considered as one oxygen molecule, to form the water vapor. So we can call the oxygen here is the last receptor for the electrons. So why we can call the Krebs cycle as aerobic respiration, although we didn't use oxygen? Because the concept of oxidation and reduction here not related for losing or gaining the gases of oxygen or hydrogen, but it's related to losing or gaining electrons by the reduction or oxidation of these molecules to transform glucose into the intermediate compounds till reaching the electron transport chain. But what will happen if the cell doesn't contain oxygen or not exposed to oxygen? It will make something called fermentation. It will start the glycolysis steps, moving inside the glycolysis steps, then it will not enter the Krebs cycle. It will make something called fermentation. And the fermentation depends on the type of the cell. If this cell is bacteria, like the yogurt bacteria or the muscles inside the human body, it will form lactic acid instead of entering the Krebs cycle. And beside the lactic acid, it will form 2 ATP molecules only. This 2 ATP molecules not enough for the muscles inside the human body to be relaxed. So the spasm occurs or muscle contraction occurs without releasing these muscles or form relaxation again. Also inside the bacteria, it will produce only 2 ATPs beside the lactic acid. So it's called lactic acid or acidic fermentation. And if the type of the cell is yeast and has no oxygen to complete the Krebs cycle, so this yeast will enter another fermentation called alcoholic fermentation, producing also 2 ATPs molecules and 2 carbon dioxide molecules and ethyl alcohol. It's called alcoholic fermentation. Till now we have finished this lesson. Thank you for listening. Hello secondary 1 students. Today we will talk about the cell structure or complete the second part of the cell structure. Let's start with the cytoplasm. As we said before, the cytoplasm is a jelly-like fluid which contains all the organelles and contains the membranous organelles and non-membranous organelles. So what is the difference between membranous and non-membranous? Membranous organelles which contains envelope or contains a membrane, and the non-membranous organelles like the ribosome and the centrosome. There are two only non-membranous organelles, ribosome and centrosome. Let's start with the non-membranous organelles, ribosome. Ribosome has two sides to found inside the cytoplasm or attached to the endoplasmic reticulum. As we said in the last lesson, that ribosomes formed inside the nucleus by the nucleolus, then transfer outside the nucleus to attach to the endoplasmic reticulum. And we have two types of endoplasmic reticulum according to attachment of ribosome or not. If the endoplasmic reticulum has ribosomes, it's called rough ER, or if it's not contain or it doesn't contain ribosomes, it calls smooth endoplasmic reticulum. So if the ribosome attached to ER, its function is to making the protein. But if it's free inside the cytoplasm or the ribosome didn't attach to the endoplasmic reticulum, its function is to make the protein of the cytoplasm. The second non-membranous organelle is the centrosome. And note that centrosome is found inside the animal cell only, it doesn't found in the plant cell. It's the structure which is responsible for the cell division and responsible for making the filaments inside the cell. Filaments like what? Filaments like the spindle fiber here, which is spread or attached between the two poles of the cell for the chromosomes to arrange it or to found to be divided. So the centrosome helps in division of the cell. So what is the structure of the centriole? Centriole is one of the components of centrosome. So the centrosome composed of two centrioles. It's like a circle, contains two centrioles. One of these centrioles composed of triplets microtubules, 1, 2, 3 triplet microtubules in 9 groups. This is the first group, second, third, fourth, and so on. So if you count these groups, these triplets, they will be 9 groups. If you multiply the 3 tubules or triplets by 9 groups, 3 by 9 equal 27. So each centriole inside the centrosome contains 27 microtubules, and if you want to calculate the number of microtubules in the centrosome, so 27 multiplied by 2. Why? Because each centriole inside the centrosome consists of 27 microtubules, and the centrosome contains 2 centrioles. Recall the function of the centrosome again. Its function is to form the spindle fibers which attach the two poles with each other of the cell for the chromosomes to attach on for the division of the cell to be completed. Number 2, it forms the flagella, which is something like the tail for the cells to move, like the euglena, which a type of microorganism in the water pond moves by flagella. Also, we can find the flagella in the tail of the sperm, which is the reproductive cell or the gamete of the male human or animal. Also, the centrosome will form the cilia. Cilia, like small hairs, surround some microorganism like paramecium. Also, cilia can be found inside the respiratory system and inside the trachea to remove all the wastes outside the respiratory system. So, to summarize, the function of centrosome is to help in the cell division by forming the spindle fibers, to form flagella, and to form the cilia. But here we are talking about the animal cell. It contains centrosome, so it can divide easily. But in case of plant cell, it doesn't contain centrosome. So how it can divide? By the cytoplasm can condenses to make the same function of the spindle fibers or the same function of the centrosome. Also inside the nerve cell there is no centrosome, so the nerve cell can't be divided at all. Also inside the red blood cells doesn't contain nucleus, so it can't divide also because it doesn't contain chromosomes. So we have here special cases for the centrosome. Let's complete the membranous organelles. We have finished the non-membranous organelles. Let's start with the non-membranous organelles, and the first organelle with us is the endoplasmic reticulum. The main function for the endoplasmic reticulum at all, whatever its type, is to transport substance inside the cell or to move substance from place to another inside the cell. It's like the tubes. But we have two types of the endoplasmic reticulum according to attachment of ribosome on it or not. If it contains the ribosome, it's called rough ER or rough endoplasmic reticulum, and its function is to making the protein. So if we asked about the organ inside our bodies that contains or the rough endoplasmic reticulum is more any organ which secretes enzymes or hormones. Why? Because enzymes or hormones, nature, most of them are formed of proteins. So the rough ER secretes proteins. So the rough ER will be increased inside the endocrine glands, which secretes hormones, or inside the digestive system, especially the stomach, which secretes the enzymes. The second type of the endoplasmic reticulum is the smooth endoplasmic reticulum. Here the smooth endoplasmic reticulum function three functions. One of them is make detoxification. Detoxification means change the toxic substance into less toxic substance, or make it less poisonous. Number two is to store glycogen and change glucose into glycogen. Number three is the formation of the lipid. So this function especially occurs inside the liver cells, which are called the hepatic cells. So if we talked about the smooth endoplasmic reticulum, which doesn't contain the ribosome function, is to found inside the liver cells or hepatic cells to make these three functions: is to formation, is to form the lipid, is to store glycogen, is to transfer the toxic substance into less toxic one. Also, the glycogen found inside the muscle, so the muscles also contains a smooth ER. Here we will talk about the Golgi bodies. Golgi bodies, also called Golgi apparatus, or inside the plant only called dictyosomes. Dictyosomes. Here is the name of it. Dictyosome. So the Golgi body is called dictyosome only inside the plant cells. What is the function of the Golgi body? Golgi body is a flat membranous sac with spherical ends. It has a role for receiving the proteins that are formed inside the endoplasmic reticulum. As you know that we have different types of proteins. We have enzymes, we have hormones, some types of hormones, and also we have conjugated proteins. So the proteins which are formed inside the endoplasmic reticulum must be modified or changed into the ready-made protein or the end form that can be transported to each part of the cell. What will make this function? Yes, the Golgi body. So each protein formed inside the endoplasmic reticulum will be transported inside the Golgi body to be classified or to be modified. So the protein formed here inside the ER will be found here inside the transporting vesicle, and transporting vesicle will go to or transfer to the Golgi body to receive them, classify them, and modify them into different types of proteins. After that, if the protein is ready, it will be transported inside other vesicles called secretory vesicles. So this Golgi body will be increased inside the glandular cells, which secretes the hormones. After that, the vesicles will be opened inside the plasma membrane to secrete the protein outside the cell to be transported in different parts of the body. Also, in some cases, this vesicle will contain some digestive enzymes and will engulf any worn out cells or senile cells or undigested food or bacteria or pathogens and lyses them or damage them or break them down. Also, they will get them outside the cell to excrete them out. So in this case, we can call this vesicles, if it contains the digestive enzymes, as lysosomes, and it can be found only in the animal cell, not the plant cell. So we can summarize the function of the Golgi body is to receive the protein formed inside the mitochondria by the transporting vesicle, modify them and make changes, then will get them out by the secretory vesicles, which will secrete their secretion outside the cell to the target cell. Here is the mitochondria, and we talked about it in the first lesson. When we were talking about the carbohydrates, the mitochondria formed of two membranes, the outside membrane and the inner membrane. The inner membrane is folded many times to increase the surface area and forming projections like the fingers called crista. If we talk about the relation between the number of crista formed and the ATP formed or the energy produced, it's a direct relation. Why? Because if the number of crista increases, it will increase the surface area, so the amount of ATP produced will be increased by in turn. And the liquid inside the mitochondria is called matrix. What about the vacuoles? The vacuoles are spaces inside the cell to store all the nutrients, the food, and wastes. Here is the last organelle inside the cell, especially the plant cell, is not found in other types of cells. We have three types of plastids: leukocytes or leucoplasts, which are called also white plastids, and chloroplast and chromoplast. Let's compare between them. Leucoplast or white plastids contains starch, as its function mainly is to store the starch, and we can find it inside the inner leaves of the cabbage and inside the sweet potato and potato. So it doesn't contain any pigment or doesn't contain any dye which give the plant any color. The second plastid is the chromoplast, and the chromoplast is the colored plastid because of presence of a pigment called carotenoid or carotenes. And this plastid will give the plant arranged between three colors: red, orange, or yellow. Its main function is to give color only. And the second type of plastid is the chloroplast or green plastid, and its function is to absorb the sunlight for the plant to make photosynthesis process and to give the plant the green color. The chloroplast has a pigment called the chlorophyll which give it the green color, and it consists of double membrane, the first one and the second one, and inside it there is a disc. Each one of them is called thylakoid. Thylakoid, each one of these discs, about 10 to 15 discs above each other, will called granum, and all of them are called the grana, the plural of them, and the liquid inside is called stroma. So we have three types of plastids: chromoplast, chloroplast, and leucoplast. Till now we have finished the membranous organelles and non-membranous organelles inside the cell. Thank you for listening.